In this activity, you're going to focus on how lateral roots or branch roots form and grow out of an existing root. You've already learned that roots don't just grow longer, they also branch, and that branching is critical for increasing surface area for absorption and anchorage. Your goal here is to use slide images of a salix or willow root to identify where lateral roots originate and to see how they connect back into the vascular system of the main root. The key tissue in this activity is the pericycle. The pericycle is a layer of meristematic cells located just inside the endodermis of the stele. Even in a mature root, these cells retain the ability to divide. This is important because lateral roots originate from the pericycle, not from the cortex or the epidermis. In other words, new roots start deep inside the root, not at the surface. As a lateral root begins to form, new vascular tissues develop from the pericycle and connect directly with the vascular tissues of the primary root. This ensures that the new root can transport water, minerals, and sugars right away. As it grows outward, the developing lateral root pushes through the endodermis, the cortex, and finally the epidermis of the original root. So even though it starts deep inside, it eventually emerges on the outside of the root. By the time a lateral root emerges into the soil, it already has its own root cap and apical meristem. That means it can elongate and develop the same regions of growth you studied earlier: division, elongation, and maturation. This process ensures that lateral roots are not just attachments, they are fully functional roots that are structurally and functionally integrated into the plant's vascular system. Before you analyze the slide images, think about this question. Why is it important that lateral roots originate from the pericycle instead of from the cortex or epidermis? Think about vascular connections, transport, and structural integration. Write your prediction in your lab notebook before continuing. Now examine the image of the salix or willow root cross-section. Start by looking at the entire cross-section so you can orient yourself. Locate the stele, the endodermis, and the pericycle. Then look for a developing lateral root. This should appear as a bulge or new structure growing outward from the main root. Using the slide image as your reference, draw and label the following structures in your lab notebook: the primary xylem, the primary phloem, endodermis, pericycle, and the lateral root. Be sure to clearly show where the lateral root originates from the pericycle and how it grows outward through the surrounding tissues. Focus on showing correct positions and relationships rather than artistic detail. Now think about what you observed in the image. First, decide whether salix is a monocot or a eudicot root. Use visible features from the cross-section, especially the arrangement of vascular tissues, to support your answer. Second, compare the new lateral root to the root tip you studied earlier in the lab. Think about what structures they share, like a root cap and meristem, and what might be different because one is a branch root forming inside an existing root. Record your answers in your lab notebook using the image as evidence. To wrap up this activity, lateral roots originate from the pericycle, which allows them to connect directly into the plant's vascular system. As they grow, they push through the endodermis, cortex, and epidermis, eventually emerging as independent but fully integrated roots. Each new lateral root has its own root cap and apical meristem, which allows it to grow and function just like the main root. Once again, this is a great example of how structure supports function in plant anatomy.